Hello and welcome to Module 1.2, the nerve impulse. I recommend, as always, to have your textbook in front of you as some of this is quite diagram heavy, particularly when we get to action potentials. Without further ado, let's get on to the reading. Think about the axons that convey information from the touch receptors in your hands or feet towards your spinal cord and brain. If the axons used electrical conduction, they would transfer information at a velocity approaching the speed of light. However, given that your body is made out of water and carbon compounds instead of copper wire, the strength of an impulse would decay rapidly as it travelled. A touch on your shoulder would feel stronger than a touch on your abdomen. Short people would feel their toes more strongly than tall people could, if they could feel their toes at all. The way your axons actually functions avoid these problems. Instead of conducting an electrical impulse, the axon regenerates an impulse at each point. Imagine a long line of people holding hands. The first person squeezes the second person's hand, who then squeezes the third person's hand, and so forth. The impulse travels along the line without weakening because each person generates the impulse anew. Although the axon's method of transmitting an impulse prevents a touch on your shoulder from feeling stronger than one on your toes, it introduces a different problem. Because the axons transmit information only at moderate speeds, varying from less than 1 meter per second to about 100 meter per second, a touch on your shoulder reaches your brain sooner than a touch on your toes. Although you won't ordinarily notice the difference, your brain is not set up to register small differences in the time of arrival and touch messages. After all, why should it be? You almost never need to know whether a touch on one part of your body occurred slightly before or after a touch somewhere else on your body. In vision, however, your brain does need to know whether or not one stimulus began slightly before or after one another. If two adjacent spots on your retina, let's call them spot A and spot B, send impulses at the same time, an extremely small difference between them in timing tells your brain whether light moved from spot A to spot B or from spot B to spot A. To detect movement as accurately as possible, your visual system compensates for the fact that some parts of your retina are slightly closer to your brain than other parts are. Without some sort of compensation, simultaneous flashes arriving at two spots on your retina would reach your brain at different times, and you might perceive movement inaccurately. What prevents this illusion is the fact that axons from more distant parts of your retina transmit impulses slightly faster than those closer to the brain. In short, the properties of impulse conduction in an axon are amazingly well adapted to your needs for information transfer. Now, let's examine the mechanics of impulse transmission. The resting potential of the neuron. Messages in a neuron develop from disturbances of the resting potential. Let's begin by understanding the resting potential. All parts of a neuron are covered by a membrane about 8 nanometers thick. That is about 1/10,000th the width of the average human hair. The membrane is composed of two layers, free to float relative to each other, of phospholipid molecules. These are chains of fatty acids and a phosphate group. Embedded among the phospholipids are cylindrical protein molecules through which certain chemicals can pass. When at rest, the membrane maintains an electrical gradient, also known as polarization, which is a difference between the electrical charge from the inside and outside of a cell. The electrical potential inside the membrane is slightly negative with respect to the outside, mainly because the negatively charged proteins inside of the cell. This difference in voltage is called the resting potential. Researchers measure the resting potential by inserting a very thin microelectrode into the cell body. The diameter of the electrode must be small enough to enter without damaging the cell. The most common electrode is a fine glass tube filled with a salt solution tapering to a tip diameter of 0.0005 millimeters or less. A reference electrode outside the cell completes the circuit. Connecting the electrodes to a volumeter, we find that the neuron's interior has a negative potential relative to its exterior. The magnitude varies, but the typical level is around 70 about negative 70 millivolts. Forces acting on sodium and potassium ions. If charged ions could flow freely across the membrane would depolarize, eliminating the negative potential inside. However, the membrane has selective permeability. This is, some chemicals can pass through it more freely than others do. Oxygen, carbon dioxide, urea, and water cross freely through the channels that are always open. Several biologically important ions, including sodium, potassium, calcium, chloride, cross through the membrane channels or gates that are sometimes open and sometimes closed. When the membrane is at rest, the sodium and potassium channels are closed, permitting almost no flow of sodium and only a small flow of potassium. Certain types of stimulation open these channels, permitting freer flow of either or both these ions. The sodium-potassium pump, a protein complex, repeatedly transports 3 sodium ions out of the cell while drawing 2 potassium ions into it. The sodium-potassium pump is an active transport that requires energy. As a result of the sodium-potassium pump, sodium ions are more than 10 times more concentrated outside the membrane than inside, and potassium ions are more concentrated inside than outside the cell. The sodium-potassium pump is effective only because of the selective permeability of the membrane, which prevents sodium ions that were pumped out of the neuron from leaking right back in again. When the sodium ions are pumped out, they stay out. However, some of the potassium ions in the neuron slowly leak out, carrying a positive charge with them. That leakage increases the electrical gradient across the membrane. When the neuron is at rest, 2 forces act upon sodium, both tending to push it into the cell. First, consider the electrical gradient. Sodium is positively charged and the inside of the cell is negatively charged. Opposite electrical charges attract, so the electrical gradient tends to pull sodium into the cell. Second, consider the concentration gradient, the difference in distribution of ions across the membrane. Sodium is more concentrated outside than inside. So just by the laws of probability, sodium is more likely to enter the cell than to leave it. Given that both the electrical gradient and the concentration gradient tend to move sodium ions into the cell, sodium would enter rapidly if it could. However, because the sodium channels are closed when the membrane is at rest, almost no sodium flows except for what the sodium-potassium pump forces out of the cell. Potassium is subject to competing forces. Potassium is positively charged and the inside of the cell is negatively charged, so the electrical gradient tends to pull the potassium in. However, potassium is more concentrated inside the cell than outside of the cell, so the concentration gradient tends to drive it out. If the potassium channels were wide open, the potassium would have a small net flow out of the cell. That is, the electrical gradient and the concentration gradient for the potassium are almost in balance, but not quite. The sodium-potassium pump continues pulling potassium into the cell, counteracting the ions that leak out. The cell has negative ions too. Negatively charged proteins inside the cell sustain the membrane's polarization. Chloride ions, being negatively charged, are mainly outside of the cell. When the membrane is at rest, the concentration gradient and the electrical gradient balance, so the opening of the chloride channels would produce little effect. However, chloride does not have a net flow when the membrane's polarization changes. However, chloride does have a net flow when the membrane's polarization changes. Why a resting potential? The body invests much energy to operate the sodium-potassium pump, which maintains the resting potential. Why is it worth so much energy? The resting potential prepares the neuron to respond rapidly. As we shall see in the next section, excitation of the neuron opens channels that allow sodium to enter the cell rapidly. Because the membrane did its work in advance by maintaining the concentration gradient for sodium, the cell is prepared to respond vigorously to a stimulus. Compare the resting potential of a neuron to a poised bow and arrow. An archer who pulls the bow in advance is ready to fire at the appropriate moment. The neuron uses the same strategy. The resting potential remains stable until the neuron is stimulated. Ordinarily, stimulation of a neuron takes place at the synapses, which we consider in Chapter 2. In the laboratory, it is possible to stimulate a neuron by inserting an electrode into it and applying a current. The action potential. Messages sent to axons are called action potentials. To understand action potentials, let's begin by considering what happens when a resting potential is disturbed. We can measure a neuron's potential with a microelectrode. When the axon's membrane is at rest, the recordings show a negative potential inside the axon. If we now use a different electrode to apply a negative charge, we can further increase the negative charge inside the neuron. The change is called hyperpolarization, which means increased polarization. When the stimulus ends, the charge returns to its original resting level. The recording looks like this. View book for diagram. On page 31. Now let's apply a current to depolarize the neuron, that is, reduce its polarization towards zero. If we apply a depolarizing current, we get a result like this. Diagram 2. With slightly stronger depolarizing current, the potential rises slightly higher again, but returns to the resting level as soon as the stimulation ceases. Diagram 3. Now let's apply a still stronger current. Stimulation beyond the threshold of excitation produces a massive depolarization of the membrane. When the potential reaches the threshold, the membrane opens its sodium channels and lets sodium ions flow into the cell. The potential shoots up far beyond the strength that the stimulus provided. And there's another diagram. Any sub-threshold stimulation produces a small response that quickly decays. Any stimulation beyond the threshold, regardless of how far beyond, produces a big response like the one shown, known as the action potential. The peak of the action potential, shown as 30 plus millivolts in this illustration, varies from one axon to another. The all or none law. Note that any depolarization that reaches or passes the threshold produces an action potential. For a given neuron, all action potentials are approximately equal in the amplitude and velocity. That is, the intensity of the stimulus cannot cause a neuron to produce a bigger or smaller action potential, or a faster or slower one. Slight variations can occur at random, but not because of the stimulus. More properly stated, the all or none law is that the amplitude and velocity of an action potential are independent from the intensity of the stimulus that initiated it, provided that the stimulus reaches the threshold. By analogy, imagine flushing a toilet. You have to make a press for at least a certain strength, the threshold, but pressing harder does not make the toilet flush faster or more vigorously. In a similar way, when you flick the switch on to turn on the lights in your room, flicking the switch harder would not make the lights brighter. Although amplitude, velocity, and the shape of action potentials are consistent over time for a given axon, they vary from one neuron to another. Thicker axons convey action potentials at greater velocities. Thicker axons can also convey more action potentials per second. The all or none law puts constraints on how an axon can send messages. To signal a difference between a weak stimulus and a strong stimulus, an axon cannot send a bigger or faster action potential. All it can do is change the timing. By analogy, imagine Morse code. You can send signals to someone by making beeps on and off at varying speeds and rhythms. For example, beep beep, beep beep, beep beep might mean something different from beep, 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 beep. And I felt silly doing that. But it better be for your education. Back to the book. The nervous system uses both kinds of coding. For example, a taste axon shows one rhythm of responses for sweet tastes and a different rhythm for bitter tastes. The molecular basis of the action potential. The chemical events behind the action potential may seem complex, but they make sense if you remember these three principles. 1. At the start, sodium ions are mostly outside the neuron and potassium ions are mostly inside the neuron. 2. When the membrane is depolarized, sodium and potassium channels in the membrane open. 3. At the peak of the action potential, the sodium channels close. A neuron's membrane contains cylindrical proteins. Opening one of these proteins allows a particular type of ion to cross the membrane. Which ion crosses depends on the size and shape of the opening. A protein that allows sodium to cross is called a sodium channel or sodium gate. And one that allows potassium to cross is called a potassium channel. The axon channels regulating sodium and potassium are voltage-gated channels. That is, their permeability depends on the voltage difference across the membrane. At resting potential, the sodium channels are fully closed and the potassium channels are almost closed, allowing only a little flow of potassium. As the membrane becomes depolarized, both the sodium and potassium channels begin to open, allowing freer flow. At first, the opening of potassium channels makes little difference because the concentration gradient and electrical gradient are almost in balance anyway. However, opening the sodium channels makes a big difference because both the electrical gradient and the concentration gradient tend to drive sodium ions into the neuron. When the depolarization reaches the threshold of the membrane, the sodium channels open wide enough for the sodium to flow freely. Driven by both the concentration gradient and electrical gradient, the sodium ions enter the cell rapidly until the electrical potential across the membrane passes beyond zero to a reverse polarity. Of the total number of sodium ions near the axon, less than 1% cross the membrane during action potential. Even at the peak of action potential, the sodium ions continue to be far more concentrated outside the neuron than inside. Because of the persisting concentration gradient, sodium ions will still tend to diffuse into the cell. However, at the peak of action potential, the sodium gate snaps shut. Then what happens? Remember that depolarizing the membrane also opens potassium channels. At first, the opening those channels makes little difference. However, after so many sodium ions have crossed the membrane, the inside of the cell has a slight positive charge instead of its usual negative charge. At this point, both the concentration gradient and the electrical gradient drive potassium ions out of the cell. As they flow out of the axon, they carry with them a positive charge. Because the potassium channels remain open after the sodium channels close, enough potassium ions leave to drive the membrane beyond its usual resting level to a temporary hyperpolarization. At the end of this process, the membrane has returned to its resting potential, but the inside of the neuron has slightly more sodium ions and slightly fewer potassium ions than before. Eventually, the sodium-potassium pump restores the original distribution of ions, but that process takes time. After an unusually rapid series of action potentials, the pump cannot keep up with the action and the sodium accumulates within the axon. Excessive buildup of sodium can be toxic to a cell. Excessive stimulation only occurs under abnormal conditions, such as during a stroke or after the use of certain drugs. Action potentials require the flow of sodium and potassium. Local anesthetic drugs, such as Novocaine, attach to the sodium channels of the membrane, preventing sodium ions from entering. When a dentist administers Novocaine before drilling into one of your teeth, your receptors are screaming, "Pain, pain, pain!" But the axons cannot transmit the message to your brain, so you don't feel it. Propagation of the action potential. Up to this point, we have considered how the action potential occurs at one point on the axon. Now let's consider how it moves down the axon. Remember, it is important for axons to convey impulses without any loss of strength over distance. During an action potential, sodium ions enter a point on the axon. Temporarily, that spot is positively charged in comparison with the neighboring areas along the axon. The positive ions flow within the axon to neighboring regions. The positive charges slightly depolarize the next area of the membrane, causing it to reach the threshold and open its voltage-gated sodium channels. Then the membrane regenerates the action potential at that point. In this manner, the action potential travels along the axon. The term propagation of the axon potential describes the transmission of an action potential down the axon. The propagation of an animal species is the production of offspring. In a sense, the action potential gives birth to a new action potential at each point along the axon. An action potential always starts with an axon and propagates without loss from start to finish. However, at its start, it back propagates into the cell body and dendrites. The cell body and dendrites do not conduct action potentials in the same way axons do, but they passively register the electrical event that started in the nearby axon. This back propagation is important when an action potential back propagates into a dendrite. The dendrite becomes more susceptible to the structural changes responsible for learning. Let's review the action potential. Bullet point number 1. When an area of the axon membrane reaches its threshold of excitation, sodium channels and potassium channels open. Then, at first, the opening of sodium channels produces little effect. Opening sodium channels lets out sodium ions rush into the axon. Positive charge flows down the axon and opens the voltage-gated sodium channels at the next point. At the peak of action potential, the sodium gates snap shut. They remain closed for the next millisecond or so, despite the depolarization of the membrane. Because the voltage-gated potassium channels remain open, potassium ions flow out of the axon, returning the membrane to its original depolarization. A few milliseconds later, the voltage-dependent potassium channels close. All of this may seem like a lot to memorize, but it's not. Everything flows logically from the facts that voltage-gated sodium and potassium channels open when the membrane is depolarized and the sodium channels snap shut at the peak of action potential.